Over to me. All right. Good afternoon, everyone. Thank you for having me here. So my name is James Kelly. I'm a PhD candidate at here at Trent. Figured I might as well come if I don't have to go anywhere. My supervisor's very happy. So I'm going to be talking a little bit about Lake Erie. A little bit. So if you guys are not from Ontario, if you don't know what it is, so Lake Erie is one of the Laurentian Great Lakes by volume. It's the smallest of them, but it's also very, very newsworthy in terms of the dynamics, in particular, revolving around algae blooms and these kinds of ideas. So if you guys read the news, you talk about algae blooms, you Google it. You Google algae bloom, you're going to come up with a picture of Lake Erie, probably from NOAA. They have these big, nice satellite pictures. So a little bit of the history of Lake Erie. So about 50, 60 years ago, blooms became very problematic in Lake Erie. They were very, very extensive, very, very disruptive to ecosystem services, these kinds of ideas. And then there was like a consortium put together to address sort of one of the main issues, which was point sources, particularly of phosphorus, and then regulating how much phosphorus goes in Lake Erie. And for a number of decades, there were marked improvements in the water quality in Lake Erie. But around 20 years ago or so, closer to 15 or 20, 10 actually, there's sort of a resurgence in algae blooms in Lake Erie. And in particular, the severity and the harmful nature of these algae blooms became different. So harmful in that they produce toxins, which are toxic to us, which we care about because it's bad. And Lake Erie is a main source of potable and drinking water for millions of people across the United States and Southern Ontario. It's very, very important. And greenhouses that have a very big industry here in Southern Ontario use the water from Lake Erie to irrigate their crops. So I'm going to talk a little bit about trying to understand these processes a bit. So part of my PhD, only probably about half of it is on this project, but it's this Lake Erie Alliance project. It's very, very large scale NSERC Alliance grant across a whole bunch of different universities, University of Windsor, Trent, University of Toronto, Queen's University, all coming together to try and better understand or trying to model the timing and severity of harmful algal blooms in Lake Erie. So we want to understand the conditions that are leading to bloom onset and in particular toxin production. So I'm not going to talk anything about toxin production because I don't know anything about it all that much. And then a main purpose is trying to develop early warning models to safeguard drinking water. A little history again. So back in 2014, there was a harmful algae bloom shown here in this photo, which is the most severe harmful algae bloom ever recorded in Lake Erie. It shut down potable and drinking water for hundreds of thousands of people across Toledo and Ohio for an extended period of days. And that has very severe consequences in terms of monetary things. And also people just need water to drink and for other purposes. So my role sort of in this project is trying to understand how environmental conditions within the lake affect the metabolic status of the lake, and then also statistically qualifying what I'm calling blooming periods, but I don't have time to talk about that sort of aspect. So I'll only be dealing with the first part. For some reason, I thought I had half an hour for this talk, and I had to cut it in half. So I'll see what I can get through. I cut a lot of slides. So just a little overview about metabolism, in case you're unaware. So we're talking about ecosystem metabolism. It's essentially just the flux of carbon between organic and inorganic states, right? And that's mediated by biology and these biogeochemical cycles. So autotrophs, these things that produce their own energy, right? They fix inorganic carbon into biomass, and then they make sugars, these kinds of ideas. Then there's heterotrophic evasion of carbon. So they're breaking down that organic carbon and releasing it as inorganic carbon, principally CO2, but also methane, these kinds of ideas. So a couple terms I just want to briefly talk about just to familiarize you. So when we're talking about gross primary production, we're talking about principally oxygen being produced by autotrophic activity, but functionally it's about carbon. We're talking about ecosystem respiration. We're talking about the consumption of oxygen and production of CO2, and net ecosystem productivity is the summation of gross primary production and ecosystem respiration. And the productivity ratio is just something is commonly defined either way, but it's just the ratio of primary production to ecosystem respiration. And these kinds of things relate to the overall trophic status of the environment. So it's important in terms of, even though it's functionally about carbon, it's much, much easier to model and to measure as a function of oxygen because with the mathematical models are much easier to run. You don't need a certain number of variables, which are really hard to have in continuous data. But it relies upon this sort of inverse relationship between oxygen and carbon. So through a diurnal cycle, for example, right, oxygen production tends to peak, and then there's a corresponding sort of decrease, so there or removal of CO2. So you have these opposing trends over time between, and you're just assuming sort of a simplistic 1-to-1 molar ratio. For every unit of oxygen that's produced, we're consuming, or the inverse for CO2. And these are what the metabolism models rely upon, this relationship. So I want to talk a little about the deployment in Lake Erie and how we're monitoring this. So we have buoys deployed in Pigeon Bay, specifically in Lake Erie. This isn't the best map I've ever made. I probably should have had another one showing Lake Erie in general. But this is sort of, if you're familiar with this area, but this Point Pelee is the point that comes down. So out there, it's a national park. It's all right in terms of that as national parks go. Then there's Pelee Island, these kinds of ideas. So this is the general spatial area that we're working in. So we had 5 buoys out there monitoring various physiochemical variables, things like, obviously we need oxygen, things like temperature. We have physical variables like wind, air temperature, air pressure, these kinds of ideas. But we also had other fluorometers deployed that measured things like nitrate in real time. They measured things like turbidity, CDOM, which is a colorized dissolved organic matter, which is related specifically in that spectrum to dissolved organic carbon concentrations, these kinds of ideas. Probably some other things I'm forgetting. Oh, and then pigments, probably one of the most important parts, pigments. So looking at measuring pigments for relating to biomass in chlorophyll for cyanobacteria and other sorts of things. And also phycocyanin, which is a specific pigment for cyanobacteria. Chlorophyll is more ubiquitous. So we ended up deploying these buoys. We deployed them every summer for the last 3 years. We're in our last typical field season. We might do another field season, which I hopefully won't be involved in. I might be finished by then. But what we do is we deploy these buoys over the summer months, so ice-free months, even though there's no ice in Lake Erie for the last couple of years. And then we also take water quality grab samples that we use to measure various forms of nitrogen and phosphorus within the water. And also carbon. So my lab's very big on carbon. So if you don't know Meg Izanopoulos, she's my supervisor. She studies carbon. She's a carbon wizard. So obviously I'm forced to care about carbon as well. So when we collect these weekly water samples, roughly we measure all of these sorts of various chemical constituents. So metabolism modeling. So I'm just going to be running through. I don't have time to walk through the actual metabolism modeling process because that's one of the things I had to cut. So I'm just going to be showing the output of it, but it was an inverse Bayesian model that is solving the most plausible representation of or explanation for the change in dissolved oxygen over time using components in terms of GPP, ER, as well as the gas sort of mixing dynamics of it. And I'll also be showing graphs looking at the cumulative NEP over time to see whether or not it's net autotrophic or net heterotrophic over time throughout this period, which is important in terms of carbon budgeting, these kinds of ideas. So just looking at this graph, so we're showing in the green is the GPP over time, so gross primary production over time from last summer. So this is 2022 specific. And then the red is the ecosystem respiration side. So it's just shown as a function of oxygen. So ER is consuming oxygen, productivity, gross primary production is producing oxygen. Then on the right are just showing sort of the correlative relationships between GPP and ER. So they have very, very strong correlations, whether or not you're looking at them by location or cumulatively pooling those together, the way you look at it. And if you just look at, so these are the 5 sites where we have the buoys where we model it for separately for each site. And you can see that there's pretty nice, or I guess complicated stuff in terms of, even though they don't vary that greatly over a sort of a spatial range, they're within 15, 20 square kilometers of each other for the most part, these sites. You're still getting these very, very site-specific peaks in metabolism. So all these graphs just look different, right? You have these peaks at different times of the year, different scales, all these kinds of ideas. So it is a very spatially and temporally heterogeneous system, even over a relatively small spatial scale, because Lake Erie is just a very complicated lake is what I'm learning. It's a lot of fun sometimes. So, and then we look at the cumulative net ecosystem productivity. So if we're just taking the net sort of summation of gross primary production and ER over time, we can look at over this time period between May and October, for across all the sites, we're net autotrophic, meaning that we are pulling more CO2 out of the atmosphere in Lake Erie than we are emitting. So it's a net sink for CO2 over that period. If you look at actual carbon flux data for the entire sort of annual period, it is heterotrophic by quite a margin. But over this period, all the models suggest that it is net autotrophic over this period. So it's producing more oxygen than it's consuming. It is fixing more carbon than it is releasing. I just want to talk about some analyses also very, very briefly. So looking at those, so using the water quality samples that we collected at each site approximately every weekly, I'm just going to show you so results from partial least squares regression, relatively simple by comparison analysis that you guys talk about. And then the simplest form of a time series analysis you can do just in terms of a Kendall rank correlation test for the in situ monitoring, that we have continuous data for certain variables over time for every day, not necessarily related to the grab samples. So if you look at the PLS biplot, so these multivariate plots are always really, really fun to break down at the start, because they're kind of weird. So PLS is just, I'm sure I don't have to explain it, but just in case someone hasn't sort of used this analysis before, it's a multivariate regression technique where it's kind of similar to PCA. It's incorporating regressions and PCA together. So you're reducing dimensionality in your data set and your predictor variables. So you're taking a large number of predictor variables that can be collinear, they can vary with each other. It doesn't really matter all that much with PLS. And you're reducing that to a smaller number of component variables. And then we're plotting these sort of in space. And we have this line that goes through with the GPP, the big sort of black line. I think it's black. I'm colorblind, so I'm guessing that's black. That's what I set it to. So that's the loading vector. So that's the relationship that GPP has with these two components that we decomposed our predictor variables with. And then we kind of relate the relationship between GPP and the other variables by its orientation to this line within the space. So you have all these other data labels there. So looking at things like water temperature, photosynthetically active radiation, cumulative rain, then concentrations of nitrogen, concentrations of phosphorus, the different fractions there, these kinds of ideas. So how close you are to the origin, which is where the two dashed lines converge at 0, 0, as well as how close you are to the line in terms of the GPP loading vector, represents the strength of the relationship, the importance that these variables have in terms of predicting GPP. But unfortunately, this was kind of a bit of a dud analysis, as can happen at times in ecology. I think everyone's been there. But the only positive important relationship that GPP had with any of these variables was actually with water temperature, which is logical because there's enzyme kinetics, these kinds of ideas. As temperature increases, the activation energy for reactions like this tends to decrease. But then there was also negative relationships, very strangely, with all of your nitrogen and phosphorus fractions. So they're kind of far away from this line. They're kind of oriented outside of this 90-degree angle of this line as well, where the intersection happens. And that's kind of a strange thing to happen, because when you have, typically when you're talking about these kinds of ideas and eutrophication, these ideas, there's positive, you expect a positive relationship. As nutrients available increase, you'd expect there to be some more metabolic activity happening, more growth, more biomass being. So I think that this relationship was just functionally limited by the sample size ended up having across all 5 of these sites. There was only about 54 observations for 54 times a week data total, not for every site, unfortunately. Because Lake Erie does not like to play nice. Even just a little bit of wind can generate significant wave action. And if you've ever been on a boat where the waves are just going up and down, a relatively small boat, it's not a whole lot of fun. So this analysis can be improved over time as we include more samples. We can pool those data together. But we do again have continuous data daily time series for certain variables that I want to talk about. And I want to see if that relationship with certain nutrients continued in that aspect. So looking at gross primary production as a function of nitrate, so we had deployed these seabird Suna nitrates, so they measure nitrate over time. And the graph on the left, I'm just showing a z-score. I just transform things into z-score because it lets me plot different time series on the same scale. Just makes ggplot work well with multiple things. I don't have to use the base plot function. But if you're showing, nitrate in this case is shown in, I think, is that purple? I think I selected purple for that color. Just vastly different colors because I do struggle with that. So you can see a general trend here of nitrogen depletion over time. That's very typical in freshwater lake systems. You would see just because nitrogen can be lost to the atmosphere through denitrification processes over time. So you tend to see this consistent pattern across all 3 of these sites where we had these deployed. And just on the right, we have sort of this scatter plot and the relationship using Kendall's correlation test. And the associated p-value. So at 2 of the sites out of the 3, there were significant negative relationships with nitrate over time with the metabolism. So that does sort of fall in line with the partially squared regression. And then the next variable I want to talk about was actually wind. So GPP as a function of the daily sort of max wind speed that I looked at. These time series are a bit more complicated to look at because there's so many plots there. But in general, you can again sort of see this discordance, what you would call with Kendall's and this rank non-parametric test, wherein that the big positive lines don't tend to interact with each other. across these two time series, and then across almost all the sites except for one, which we can say weakly or weaker evidence for it, but the p-value was significant across all the sites, and that there was a negative relationship between wind and primary production. So as wind speed, which impacts mainly the mixing dynamics in the lake, increases, primary production decreased. And then looking as a function of precipitation, this is actually a very interesting variable, something I noticed. So even though if you look at all the scatter plots and the p-values, none of the relationships across all of these sites are significant. So there's no relationship between GPP and the 7-day total precipitation. And then if I look at the same thing with respect to ecosystem respiration versus the 7-day total precipitation, again, there's no significant relationships. But if you take the ratio of the two, so looking at representing the balance of that ecosystem, the metabolic processes, you do get significant relationships. So even if there isn't necessarily a significant relationship between each of these processes and precipitation, it still can significantly affect the balance of those two processes, whether or not the ecosystem is autotrophic at a certain time or heterotrophic at a certain time, which is kind of interesting. I'm still trying to figure out, my supervisor wants me to write this up and do a paper. I'm trying to figure out how to justify that, which is proving fun. And then sort of looking at it as a function of the pigments, which are related to the biomass or overall amount of cyanobacteria or chlorophyll sort of in the system. We look at just at two of these sites this time, we can see sort of these more substantial overlaps in terms of the timing of the peaks in phycocyanin and gross primary production. At least at one site, site 6 is quite observant. Site 7, I still haven't decided whether or not that's those data are trustworthy in terms of that really strange bubble that you see in phycocyanin. It's a very weird, we're having a lot of sensor issues in some of these projects, which is why there's a different number of sites shown for different variables, these kinds of ideas. But regardless, there are significant positive relationships here. So generally speaking, there is a relationship between GPP and daily phycocyanin. When one is higher, the other is higher as well. And that continues into daily chlorophyll as well. So chlorophyll, another photosynthetic pigment, very, very important. It's ubiquitous across photosynthesis, phycocyanin specifically regarding cyanobacteria. So again, you can see at two of these three sites, you have significant relationships between GPP and mean daily chlorophyll. So that's because there's overlap in there in terms of the GPP, the metabolic balance that you're seeing. It provides some evidence that there's some in these particular sites that these sensors are working as we intended them to do. And then some very basic conclusions that I just want to talk about. So the metabolic statuses do not impact pure nutrient driven, but I can only say that with respect to nitrogen specific nutrients. We don't have a continuous time series of phosphorus, which is a far more important limiting variable with respect to growth. You need phosphorus in order to grow. In terms of biomass, there were actually stronger associations either with the underlying metabolic processes themselves or balance or of the two with the actual underlying physical processes. So this suggests that the physical stability of a system is important in terms with respect to the metabolic balance here. There's some other things I'm working on now in terms of the mixing dynamics of Lake Erie as well, looking at things like bronchophysalia, Schmidt stability, these kinds of ideas that are representing the thermal stability of the water column and how that relates to the metabolic processes. But that's still ongoing. I didn't have time to finish that. My supervisor, she asked me working on a lot of different things. And then just I like to always good to acknowledge sources of money and help. Those are things you can never get enough of, I guess, in academia for sure. And with that, I'm happy to answer any questions. Thank you for your attention.
Let's leave questions for the break because we have 4 talks to get through, and unfortunately, we just don't have a lot of time for each person.
I tried to condense it down as much as I could.
Not you at all. It's me. I crammed everybody in, and we should have had parallel rooms, but then there'd be even less people to talk to, and that's even less fun. All right, back in Zoom. Giuliani, are you there? Or yes, hello.
Yes, can you hear me?
We can hear you. I'm just going to highlight you. There we go.
Okay,
so we can
that's
see
fine.
you and share your content up to full screen.
Yes.
And let's see, we're now going, everyone, from water and contaminant things to air and contaminant things. So we're going to talk a little bit about pollution risk in the air. Over to you, Giulia.
Oh, yes, let's move to introduction. So we all agree that degradation of air quality due to anthropogenic activities poses a serious challenge to human health, and relation between air quality and health has been widely discussed and assessed, especially in medical literature. We know that ambient air pollution is a real problem in cities throughout the world, in particular in the mega cities of developing countries. Therefore, it's of extreme importance to be able to measure health risk related to air pollution. So how can we reach the purpose? At monitoring sites located in urban, suburban, and rural areas, we observe concentrations for measure ambient pollutant. And in order to measure air quality, this concentration must be combined into a single air pollution indicator. But the combination procedure is not just straightforward, because it should reflect the dependency behavior among pollutants. As we know from finance risk studies, any risk assessment procedures should consider correlations among relevant variables. But a correlation analysis of pollutants of pollutant variable based on common correlation measure fails to capture the various properties of air pollution data, such as dynamic changes over time. Therefore, the need for reliable estimates of dynamic correlation has motivated much academic and practitioner research. In our study, we use the approach of Engel 2002. We use a multivariate Garch dynamic conditional modeling approach that allows conditional variances and covariances to be time varying and depending on past information. And this class of models has proven to be very useful in studying volatility dynamics and spillover effects. The MGARS DCC model is computationally rather simple and can be estimated in two steps. In the first step, we account for the conditional heteroscedasticity, and in the second step, we capture the dynamics in conditional correlations. So to be more precise, what we do is, borrowing from financial risk analysis, our study proposes a combination of asymmetric MGARS DCC models, a measure of risk, to analyze the time-dependent relation between particular matter of dimension 10, nitrogen dioxide, and ozone. And our analysis reveals that there is an evident correlation that changes over time among our pollutants. So, as I just said, we analyzed data related to particular matter 10, nitrogen dioxide, and ozone, which are mandatory pollutants for calculating any background pollution index, as suggested by the Environmental Agency. And for our purpose, we have the availability of daily data covering a period of 2 years, 2014-2015, at an urban monitoring site denoted with Trento PSG. As we can see from the figure, Thor recorded using the same measurement unit, which is microgram over cubic meter. The average daily concentrations differ both in terms of value ranges and in terms of seasonality. As we can observe, there is a clear seasonality pattern in ozone, and there is also a seasonal pattern not so clear in nitrogen dioxide. So in order to transform the problem of different, in order to solve the problem of different range values, we transform our daily concentration for each pollutant into comparable index values denoted with PIKT, values which vary in the range of 0 to 100. And for this purpose, we use a linear interpolation of the reference scale values, as in Morena 2004. So we use this formula where XKT is the actual pollutant concentration, and BPH and BPL are the breakpoint or threshold values established by the environmental agency for classifying pollutant values with respect to health risk classes. Our breakpoint for our index values are denoted with PIH and PIL, and these threshold values are 25 for good air quality, 50 for low pollution, 70 for moderate pollution, 85 for unhealthy for sensitive groups, and 100 for unhealthy. So we have got new health risk classes. But as we can see from the figure, our new time series show a problem that is related to the stationarity of the new time series. As we can see, there is a clear non-stationarity, in particular in the mean level of the transformed data. So now we have to solve the problem of non-stationarity, and for treating this problem, we use a model suggested by Harvey in 1989. So we are going to estimate the unobserved trend component of our series using the following model, where mu t denotes the local trend level, Bt is the local trend slope, and eta t and t are IID normal errors with mean zero and constant variances. And this is a model designed for non-stationary time series that shows second order stochastic trends. So using this procedure, we can get the detrended index time series. And as we can see from the figure, this new time series, this detrended time series, look rather stationary. So they show some time varying variability and also some volatility clustering in their variability, especially in this period at the beginning of 2015. So this is quite interesting. I mean, this figure is quite interesting because we see these three time series which look like financial returns. They are very similar to the behavior of financial return. That's the reason why we have chosen to use a methodology which is largely used for analyzing financial time series. And the methodology we are going to use is the following. So we start with a general specification of the model for the time series, that is a vector autoregressive model of order 1 for the conditional mean of the vector of the trended time series, denoted with YT, and specify the innovation epsilon t as given by the product of the square root of matrix HT times some new disturbances, some new innovations denoted with VT, where HT is the covariance matrix of the innovation epsilon t conditional on information given at time t minus 1. So oh, sorry. What does that mean? Can you hear?
Yeah, it's okay. Down the bottom, there's a resume button if you want.
Yeah, I have just moved the cursor in. Okay, so let's go back. So, and in Engels' approach, in Engels' 2002 approach, the focus is on the separate modeling of the conditional covariances and of the conditional correlation. So in the first step, we estimate the conditional variances. That is, for each variable, we estimate its conditional variability using a different Garch model specification, which can include possible asymmetric effects of innovation. Then the conditional covariance matrix HT is decomposed as follows, where the square root of dt includes the conditional volatilities estimated in this first step. Then we move to the second step, and the second step consists in generalizing Boller's constant conditional correlation model to capture dynamics in the correlation, and hence the name dynamic conditional correlation. And the correlation, the dynamic correlation matrix RT, as we can see here, is not explicitly driven by a dynamic model, but is derived from the standardization of a particular matrix, which is QT, which is a symmetric positive definite matrix characterized by a dynamic structure. Oh, sorry. And several specifications have been suggested for QT, and the specification that we have chosen is the following, which is similar to a Garch 1-1 model, where epsilon tilde represents new standardized innovation, and parameters lambda 1 and lambda 2 are non-negative and govern the dynamics of conditional correlation. And in order for the model to be mean reverting, the parameters must satisfy the condition that their sum is between 0 and 1. As I said before, Garch DCC model have gained popularity because of their computational tractability. And in fact, the parameters can be estimated using this two-step approach, unlike, for example, Garch BEEK models. So what we get from our analysis is represented in this feature. As we can see, for each pair of pollutants, we have estimated their time-varying conditional correlation. In the upper left of the panel, we have the conditional correlation between PM10 and NO2. Here in the right upper of the panel, we've got the dynamic correlation between NO2 and O3, and this is between PM10 and O3, and this is just the same graph in a unique graph, represented together in a unique graph. And so So the conditional, I mean, these time series, which are the conditional correlation, as we can see, they vary a lot over the period of observation, emphasizing the importance of computing the conditional correlations, whereas the unconditional correlation would be not significant over the same period. And what should concern us most are the positive correlations, while the negative correlations are signals that pollution condition are less worrying. So as we can observe, if we look carefully, we can observe an upward trend in the correlation between PM10 and NO2, and a clear seasonal pattern in the correlation between PM10 and O3. And moreover, there are several peaks in the correlation between NO2 and O3. Overall, we can see that dynamic conditional correlation peaked and reached high positive values, particularly in summer 2015, as well as in the first half of 2014. So this is a clear indication that there are spillover effects between pollutants, and when measuring pollution, we should consider them. Therefore, we should look at the combined effects between pollutant and not at the single pollutant effect. So this is just to show you how we can use a financial model even for computing a value at risk for pollutant. In this figure, we report estimated 80% values of risk for the index time series of PM10. That is, it's 80% quantiles, but this is just how we can still use financial literature, financial methodologies, but as we have understood, for calculating appropriate value at risk for a combination of pollutant, we must consider possible spillover effects and co-movements among pollutants. And these tend to rise risk above and beyond levels purely justified by single pollutants. So these are just few conclusions that I have already discussed during the presentation. And so that's all.
Thank you very much. I've worked on some similar problems. Yeah, it's a very tricky problem.
Okay, sorry, I must leave you because university is closing at 9 o'clock.
Yes, it's very late. Go home.
Yeah, in Italy it's very late.
So late in your Friday, and have a wonderful weekend.
Okay, thanks to you too. Bye-bye.
Bye-bye. All right, our next speaker is local. Again, I believe, Pandita, right? Yeah, you're up. Sorry, you just gave me a look like I just said the wrong person. I'm like, did I actually just choose the wrong person?
It's me.
Is this when the JP? Or where did
No,
you
it
talk?
was in downloads, I think.
Okay, let's just double check. Yeah. That one. Okay. Perfect. And we'll go full screen. And then I just need to do one last thing over here. It's not here, sorry. It's this. It's probably, there we go. Just give it a second. That will go up. Okay.
Thank you.
Thank you. I'll hand it over to you.
Hello, everyone. I'm a PhD student here at Trent, and I'm presenting some of the results from an ongoing study that we are conducting in Lake Superior, looking at metabolism patterns using autonomous underwater vehicle data. So as my colleague James mentioned, we are interested in looking at metabolism in lake ecosystems. just simply metabolism of an ecosystem looks at, quantifies the amount of organic carbon that all the organisms within the ecosystem produces through photosynthesis, which we call as cross-primary production. And then the amount of organic carbon that is being utilized for respiratory activities by all the organisms, that is called as ecosystem respiration, and balance between these two processes, that is called as net ecosystem production. This metabolism of an ecosystem gives us a comprehensive idea about the energy flow within the ecosystem, nutrient cycling, productivity, and overall health of the ecosystem. That's why we are interested in this aspect. In aquatic ecosystems, rather than quantifying organic carbon produced and utilized, we quantify the amount of oxygen produced and utilized, because the stoichiometric proportions of organic carbon and oxygen involved in these processes are equal, and it's very easy to quantify oxygen. So in this process, we quantified the dissolved oxygen in water body time series data more than 24 hours, along with the water temperature and many other variables to inversely model cross-primary production and ecosystem respiration. The metabolism, lake metabolism studies of small to medium sized lakes have shown that many lake ecosystems have higher ecosystem respiration, meaning that they emit carbon dioxide to the atmosphere. We don't know whether this is true for many large lake ecosystems, because we have very less number of research in large lake ecosystems, because we have to set up these sensors or buoys that collect fine temporal data in this large lake ecosystems, which is very expensive and labor intensive. So we have very limited knowledge on large lake metabolism. For instance, in Lake Superior, we have lake metabolism studies that are conducted in a very small area, or we kind of get an idea about metabolism using these satellite-based model productivity studies or from paleoproxy studies, which is not what we want. So to fill this knowledge gap, we can use data collected by these autonomous underwater vehicles, which are known as gliders as well. These devices are mainly used in marine studies, but they can effectively use in large lake ecosystems as well. Unlike those stationary buoys or sensors, these devices oscillate in the water column in a zigzag pattern while continuously collecting water quality variables. We can program them to go on a specific area of the lake for specific number of days. And most importantly, all the data points have a specific location, time stamp, and a depth correspondent to it, which is very useful in metabolic studies. So the consortium of Great Lakes gliders have directed many glider missions in Great Lakes, and most of them are done in Lake Superior. As you can see from this slide, they have done many glider missions, about 17 glider missions from 2014 to 2021. Most of these missions are done in summer months, with mostly in the southern Lake Superior towards southwestern of the lake. So a goal of our study was to estimate metabolism in Lake Superior using this existing glider data. As a starting point, we wanted to look at the metabolism of the upper mixed water column, which is known as epilimnion. In lake metabolism, most of the time, we measure metabolism in this upper mixed water column, because as you can see in this diagram, in summer months, where the metabolism is very high, the water column stratifies into different layers. In this epilimnion, it receives the highest amount, it has the highest amount of water temperature, and it receives the highest amount of sunlight, which is needed for the metabolism. So most of the studies look at epilimnion, and we also started from there. This slide kind of gives you an idea about how we model metabolic variables like gross primary production and ecosystem respiration from the variables we collect. I'm not going into detail. What I want to tell you is the amount of dissolved oxygen in water is balanced between biological activities like gross primary production and ecosystem respiration, and some physical processes like a gas exchange between atmosphere and mixing within the water column. So we filter out the dissolved oxygen that is result of this biological activity. And then we take into consideration that during the daytime, the gross primary production changes, and gross primary production happens only within daytime because it needs sunlight, and it changes throughout the day. And we assume that ecosystem respiration is constant throughout the day. Then the data collection, as I mentioned, I used glider data. I downloaded glider data from my iOS glider app. This is open for anyone can download this data. Then after downloading this data, I filtered out the epilimnitic data using temperature of the water column. And this is just to give you an idea about temperature profile that was developed for one typical summer day, how it would look like. So I only filtered out data in this upper top layers. I didn't use the other layers. And then using the spatial location, I added wind data from National Data Buoy Center. These are the wind stations I used for my study. So depending on the movement of gliders, I added 2 to 3 wind stations. And for some of the glider missions that are long and spread out, I added about 7, 8 wind stations. Then finally, I modeled irradiance, the light data in R using spatial location and time of the day. And this is to show you another aspect of how glider data would be like. As we collect this data from gliders, as they move through the water column, there is a slight delay in data recording in these devices. These are data from 2 ascending and descending profiles that are close to each other. You can see they are not the same. At one depth, they should have the same dissolved oxygen concentration, but it doesn't. So we have to, for each glider mission, we have to find out this offset time and correct our dissolved oxygen and temperature. Then only we can use it for further analysis. And then we modified and adapted some of the methods from this paper I mentioned here, study done by Baroni and colleagues. They've used this glider data to estimate metabolism in marine environment. Moving to the results, this is a summary of gross primary production and ecosystem respiration values I estimated using the glider data based on months of the year, and I've used many years of data. So without getting into all the specifics, what I want to mention is the values that we estimated for gross primary production and ecosystem respiration are quite low for when you compare with many other lake ecosystems. This is not surprising given that Lake Superior is a clear lake, less productive lake. If you look at the nutrient values, they have very low nutrient concentrations. So the values we reported are justifiable looking at the nutrient data for Lake Superior. And this graph shows the ratio between gross primary production and ecosystem respiration. So this red line shows the 1-to-1 ratio, which means the amount of organic carbon produced within the ecosystem is being utilized, the same amount being utilized. But you can see many of our values are below this red line, meaning that ecosystem respiration is higher most of the time for Lake Superior, meaning Lake Superior also emits carbon dioxide to the atmosphere most of the time, just like small and medium-sized lakes. And then net ecosystem production, which is the balance between gross primary production and ecosystem respiration, is shown here. So again, this red line, the 0 means the system uses the same amount of carbon that is being produced. As you can see, many of our values are in the negative region, meaning ecosystem respiration is higher, giving us a mean value in the negative region. So if we compare our values with a previous study done in Lake Superior, Sterner study done in 2010, they did a metabolism study in Lake Superior, but in a very small region, using 2 weeks of data in, I think, 2009, July. So they recorded a positive NEP value, meaning Lake Superior is absorbing more carbon dioxide. We think this difference in our value and Sterner study is because unlike Sterner study, we used many months of data, many years of data, and a spatial larger area, which increases the variability of data, which comes closer to the actual scenario. Whereas in Sterner study, they used a very small region and very few days. So, so far I've talked about the epilematic metabolism, the upper mixed column. So, and moving on to the next steps, we want to look into the deeper layers of water, specifically metalimnion. I've shown you this graph in my methods. This is a typical summer day temperature profile developed from glider data. So if I develop the oxygen profile for the same day, it would look like this, which is very different from another typical lake, because normal lakes show the same pattern as temperature for dissolved oxygen, whereas in our case, you can see there is a high dissolved oxygen concentration in the metalimnion, which shows us there's a higher amount of biological activity or metabolic activity going on in this region. Again, as I mentioned before, Lake Superior is a clear lake. The sunlight penetrates well below the epilemion, and most stratified lakes have higher amount of nutrient concentrations in the metalimnion. So although the temperature is not ideal, it provides other variables that are ideal for metabolic activities. So we are planning to estimate the metabolism in metalimnion and dig deeper. In conclusion, autonomous underwater vehicles provide a great tool in estimating lake metabolism in large lake ecosystems that can look at many time scales and integrating many depth layers. Having said that, we have this challenge of separating spatial and temporal variation, because we collect data from a device that moves through time and space. So to address this part, we have to have separate glider missions that are looking at the specific spatial, specific area or conducted during same period of the year. We are planning to do that, but in near future. However, the amount of information we can gather from the existing databases are ever increasing, and it's open source. So I think it's important to leverage this information into understanding biogeochemical processes of large lake ecosystems, which is otherwise very difficult. With that, I'd like to acknowledge all the collaborators and funding agencies, and thank you. Stop share. Stop share.
That's everything Maggie's attached to.
Yeah.
Yeah. I see CFI and CRC and all the rest of it. That's great. Yes, they cost, yes. That's, yeah. Okay, so we have one final speaker in this section now. And I'm trying to see if, Ala, are you here? And
Yes, I'm here.
okay, wonderful. So I'm showing you, yeah, under Sylvia. Okay, perfect. I'm going to spotlight you.
Okay, can I hear you?
We can hear you. Can you hear us okay?
Yes.
Okay,
Okay.
please go ahead and share your screen with us, and we'll have the final talk of this session.
Thank you. Thank you. Okay, good afternoon, everyone. This is Sylvia, a master's student at Department of Statistics, Faculty of Economics and Political Science, Cairo University. This presentation entitled A Spatio-Temporal Statistical Downscaling Model for Combining Spatially Misaligned Maximum Temperature Data using our INLA is a part of my thesis project, which is a joint work with also Amira Al-Ayouti and Abdel Nasser Saad. First of all, as we see in the map, that the area of our interest is the region of the Nile Basin countries in Africa. So the question here is why studying the climate in this region is important. Actually, it's important for many reasons. The first one, because many scientists mention that the Nile Basin countries are highly vulnerable and less resilient to the impact of the climate change. And this is due to many reasons, such as the countries that's poor dependence on the technology, as well as most of the countries' economies are particularly depends on agriculture, which is one of the sectors that is highly affected by any change in the climate. Also, for the second reason is that extreme weather events have been occurred more frequently than before, and that led to severe consequences in the Nile Basin countries, such as floods, rise in the sea level, change in the Nile river flow, and etc. Also, for the third reason, that based on the previous literature, it's notable that there is a gap in the literature in analyzing the climate variations in this study area. And this is maybe due to limited accurate data available. Last but not least, this is the area where my home country belongs. So as we read in the title, that our variable of interest is the monthly maximum temperature in the Nile Basin countries from the year 2011 to 2020. And we obtained the maximum temperature from two different data sources, as we see in the slide. The first data source is the simulated monthly maximum temperature data generated from a physical climate model with a 44 by 44 kilometer resolution. The data are downloaded for the domain of Africa from the year 2011 to 2020 at grid cells, which are found at Lattice Data, while the second type is in situ observations, come from 52 meteorological weather stations located in the Nile Basin countries from the same study period as shown in the right map. Here are the red points, which are the locations of the available weather stations. And this notes that both data sets have its own characteristics. For the first data set, the simulated monthly maximum temperature data, they are deterministic data, unreal observations that are far beyond the reality and do not account for uncertainty. While the second type are real and accurate observations that's accounted for uncertainty measurements. On the other side, the simulated monthly maximum temperature data are downloaded as in a complete coverage in space and time, while the in situ observations suffer a lot from high percentage of sparse data in space and time. Here, for example, country like Eritrea, here, which is above Ethiopia, has no available weather station at all. Country like South Sudan has only one weather station available, and so on. So the idea here is that we think to improve the production of the monthly maximum temperature data by getting benefit from the advantages of the two types of data and to overcome their limitations. But here, they are latest data, the simulated monthly maximum temperature are latest data, while in situ observations are downloaded or are measured at specific longitude and latitude. So both of them are or suffer from spatial misalignment problem. So the main objectives of this study are to adjust the physical deterministic simulated data with real in situ observations and combine both data to obtain more accurate prediction of the monthly maximum temperature along the entire spatial and temporal domain associated with their uncertainties. And this can be done through fitting a spatio-temporal statistical downscaling that can be or their outcome is a prediction at a spatial point. Thus, the model is explained in the next slides. The idea of a coordinalization model or a joint model is used to fit a spatio-temporal statistical downscaling model. Here, the idea of the joint model is to assume a bivariate or a joint distribution for both the covariate and the response, not the response only. And actually, this has an advantage over using a univariate model such that the uncertainty in the covariate could be feed forward and propagate into the model for the response estimation, as well as the covariate can be predicted at any point in the spatial domain, so the biases due to measuring the covariate and the response at different locations are accounted for. Here is the response of the model, the observed monthly maximum temperature data, and the covariate is the monthly simulated maximum temperature data simulated with 44 by 44 kilometer resolution. This explains the model specification. Here, Z1ST is a true spatio-temporal process for the simulated monthly maximum temperature data with a spatio-temporal structure correlation. And Z2 is considered as a discrepancy spatio-temporal error component between the simulated monthly maximum temperature and in situ observations. Epsilon 1 and Epsilon 2 are considered as a white noise, follows IID. Beta 1 is considered as a calibrating parameter that adjusts the simulated monthly maximum temperature process with the in situ observation, and beta 1 Z1 is like a calibrating process, while beta naught is the intercept. Thus, in our model, we have two spatio-temporal latent fields with separable spatio-temporal covariance structure, such that we assume each area has a spatial covariance with zero mean matter covariance structure and autoregressive of order 1 temporal structure. But after fitting the model, we noted that still the Z1 and Z2 cannot be able to capture all the temporal variations, and there is still a trend in the residues. Thus, we add two extra terms, which are RT and ST, that detects the trend in the temperature along the 10 years period, the RT, and ST captures the seasonal pattern of the temperature within each year in the study. The model is fitted under 3 levels of hierarchical Bayesian model paradigm using INLA. In the first level, the likelihoods of the response and the covariate are specified such that here we are talking about monthly maximum temperature data. So X and Y follow Gaussian distribution with mean psi j and eta I and sigma square X and sigma square Y. At the second level, the spatio-temporal latent field components are defined such that Z1, ST, and Z2 follows a multivariate Gaussian process with zero mean and separable spatio-temporal covariance structure. That sigma S is a matter covariance structure defined by the sigma, the variance, and the range, and sigma T is AR1 or autoregressive of order 1. At the third level, all the prior distributions of the parameters and hyperparameters are specified, such as sigma square X and sigma square Y. The reciprocal of sigma square X and sigma square Y follow log gamma, half and a half. Here we assume a perfect linear relationship between the simulated monthly maximum temperature data and in situ observations. So the pair distribution of the beta node follows a normal with 0 and variance 100, and beta 1 follows a normal with mean 1 and beta 100. Also, all the parameters that represented the matter covariance function and AR1 are specified in this level. Here is some results or some figures from the model outputs. There is the posterior mean for the trend component RT on the left, and figure on the right represents the posterior mean for the seasonal component and their 95% credible intervals. And we can note that most of the temporal variations are captured in the seasonal component. And for the trend component, it highlights that there is a slight increase in the maximum temperature from the year 2013 to 2016. That was agrees with what WMO reported, that in 2016 El Nino event occurred, which was responsible for the highest global temperature level. Then we fitted a spatio-temporal GAM model and use it as a benchmark in order to compare our model with the spatio-temporal model. In the spatio-temporal model, we fitted and using only one data type, which is in situ observations. And we ended up with the following results. Depending on the measures of model's adequacy, we ended up that all measurements tends to the spatio-temporal downscaling model led to better prediction for the data. Here is a figure that represented the monthly maximum temperature observation from in situ observations, indicated in the colored circle, over the predicted values, which seems that the predicted values have the same spatial distribution like the in situ observations. This figure for only 1 month, which is July in 2011. After that, the data were split into 2 data sets, the training data from the year 2011 to 2019, and we used the data of the last year, the year 2020, for a validation set. And here, a figure that represented the difference between the predicted values of the maximum temperature in year 2020 that represented in the blue lines over the observed in situ observations for selected 4 countries. The red lines represented the real in situ data. And it seemed that the model sounds good to capture the trend of the maximum temperature except for the highest or the lowest values. Also, the model can predict the monthly maximum temperature data better in countries that have more homogeneous temperature rather than has much variation. Just to sum up what we have and what we concluded, we can end it up with that the model can capture all the spatial and temporal dependencies in the data, as well as integrate the climate data from different sources would improve the prediction of the maximum temperature across the space and time. Okay, what about the model's limitation? Actually, as we see that the model performance was poor at predicting the temperature at the tail of the distributions, which means at extremely high or extremely low values. So we think to overcome this problem that we use the quantiles instead of the mean in order to solve this problem. Also, the model performance gives better predictions for the temperature data for countries near the equator and that have more homogeneous monthly maximum temperature data rather than countries far from the equator, such as Egypt, which leads to a problem in stationarity assumption of the spatial processes. And we think that how to relax this assumption in the model. And now we are working on this idea that relax the model to allow for a spatially varying coefficient model. And instead of using beta naught and beta 1, we are using now beta naught S and beta 1S. Finally, what about for next? Number 1, the model can be extended to allow for a multivariate models that include other meteorological variables such as precipitation. As all we know that the analysis of precipitation is also important for this study region, specifically the region near the equator line. And also we think about that we extend the model in order to allow for the predicted values or the outcome of the model to be used as a covariate in order to study the impact of the climate and its changes on many aspects in our life, such as health, agriculture, rise in sea level, the change in the river, in the Nile River, and so on. Thanks so much for your listening and attention, and waiting for any questions.
Oh, sorry, we were muted. Let's do that again for her. We had our microphone muted. Sorry about that. So do we have, we're going to take, I think, let's take 10 minutes. We'll push the next session just by 10 minutes so everyone can grab their copy. And do we have any questions for Sylvia? Because she is online here. Anyone have anything they want to ask about the work she was doing on these temperature models? And anyone online? I thought maybe I saw a hand, but maybe I was just missing that. No. I'll ask you one question. How did you find the process of learning to use INLA? Because I've used it a few times and I found it, at least my student claims it's very easy. I found it somewhat challenging to get started with. Did you find the transition into the modeling structure to be okay and you were able to translate the ideas you had about how the data should connect into those models to be straightforward?
Okay, I think you asked me about that I can detect any change in the temperature.
No, it's more just the process of using INLA, because you're using INLA software, right? So you had ideas about how those different variables should connect and you built your mathematical model, did you find the implementation to be straightforward?
Yeah.
Yeah. Okay. I had two students who had the same. They said it was very, very easy, and apparently it's just me. So that's great. Okay. We don't have any other questions for Sylvia. So thank you so much, Sylvia, for joining us today. And we are going to take a short break now, just to get everyone grab coffee and maybe have a biological break. And then we'll resume with our final session of the conference in 10 minutes. Thank you very much, everyone.
Thank you. Thank you.